This data taken from the Shandong Children Eye Study, we can see that in 4 to 6 year olds measured without cycloplegia, there's quite a high prevalence of myopia. In contrast, if they're measured with cycloplegia, it's very much lower. And you can estimate that at least 80% of the children classified as myopic without cycloplegia are in fact not truly myopic. Move to the end of schooling, the age of 17 to 18, and the figures are much more similar, and on either measure there's clearly an epidemic of myopia, but there's still about a 10% difference in prevalence. Now this makes a major difference, because if we look once again at the 4 to 6 year olds, the difference between the cycloplegic and non-cycloplegic data is made up by what are called pseudomyopes, and these are children who are not truly myopic. I've already defined pseudomyopia, those who are myopic without cycloplegia but are not myopic with it. Now it's important to recognise that pseudomyopes are not just some sort of myope or some sort of pre-myope. There's something quite distinct. Pseudomyopes do not need glasses for good distance vision, and thus the need for correction is overestimated without cycloplegia. They're equally not the same thing as pre-myopes. They're not necessarily those who are most at risk of becoming myopic. So you can ask the question, how useful is non-cycloplegic refraction? Well, it is recommended in China for school screening. This is not a bad thing because school screening needs to detect all children with myopia, even at the cost of over-referral, which can be sorted out by clinicians and cycloplegia. It can also be used for population surveillance, since changes in prevalence can be detected with this method, provided the methodology does not change. However, it's not really useful for scientific purposes or for precisely defining parameters such as the prevalence of myopia. For this purpose, you need smaller, statistically valid samples measured with cycloplegia. For this reason, we actually need a lot more information, even within the confines of the Asia-Pacific region, which is covered by our society. I've classified all the member societies of the Asia-Pacific Academy of Ophthalmology into high, moderate, or low myopia based on 3 parameters. Data on the prevalence of myopia in children completing high school, where it exists. Development of the education system from official enrolment statistics, the logic being that if only a low percentage of children complete high school, you're unlikely to have an epidemic of myopia, although many of those completing high school may still be myopic. And the third parameter I've used is performance of the educational system from PISA, because if you don't have a high-performing system, it's unlikely that you will have an epidemic of myopia. Now, there's really no doubt about those in the high category that have a high prevalence of myopia. We could do with more data from Japan and South Korea in particular, but no doubt at all. In the moderate category, what I'll call 20 to 40% at the end of schooling, we have a few countries. You'd be sure about the data from Australia, but the data from the other parts of the world is much more limited. It's so limited that it may be that Vietnam and Malaysia are heading into the high category, but we don't have enough data to know. When we look at the bulk of the members of APAO, we have very little data on none on some and very little on most. But they're all likely to be in the low category, which I mean less than about 15% at the end of schooling. There are a couple that might be making it into the moderate category. India is a very complex situation because it has a large population which is not receiving a lot of education and a highly educated sector of the population. In that, myopia might be a problem, and it may be heading into the moderate category. This might also be true for Sri Lanka, but we just don't have enough data to be sure. Despite this generally pessimistic view that I've presented, there have been some positive developments in our understanding of myopia, even despite non-cycloplegic refractions. One of the things we've discovered, particularly from data in China, is that the increase in myopia during the school years is due to educational exposures, almost exclusively, and not to increasing age. I'll illustrate that with some data from Guangzhou that I've analysed with my colleague Ding Xiaohu, but I've seen at least 3 other data sets from China that make the same point. The logic is the cut-off date for starting school in China is very rigid. If you turn 6 on the 1st of September, you start primary school. If it's later than that, you start the next year. And so you can have children in different grades of primary school who differ in only a few days in their birth date. Within a grade, there's very little difference in the prevalence of myopia or mean spherical equivalent by age. This is grade 1 students in Guangzhou, from the youngest to the oldest. You can see very little difference, although the age range is 1 year. The same is true in grade 2, grade 3 and grade 4. But if you look at the difference between grade 1 and grade 2, which is also a gap of 1 year, you can see that that additional year of schooling makes a big difference. I've tabulated down here 0.2 diopters within, 0.3 diopters between. And what the only possible way of interpreting this is that it's the exposures of being a school child that drives these changes in mean spherical equivalent. Now, this has some major implications. It means that these shifts, annual shifts across a school year, give us a cumulative measure of the impact of all the individual risk factors for myopia, even if we can't measure them. And this actually gives us a very simple measure for assessing the efficacy of myopia prevention measures within school systems. The other area where we've made some progress is in the impacts of COVID lockdown on changes in refraction. There's been a lot of concern that because children during COVID lockdown are deprived of time outdoors and may spend an enormous amount of time online, that there could be major shifts in refraction as a result. I think the best study that exists so far comes from Wang et al., published in JAMA Ophthalmology. And what it tells us is that for children in the final year of preschool, the impact of lockdown was a negative shift of 0.32 diopters. It's much the same for grade 1 students and grade 2, but then it progressively drops off. The authors suggest that this might be due to a lesser sensitivity or a greater sensitivity of younger children to environmental risk factors. I'd suggest that there's a different and possibly more plausible explanation, and that is that by the time children have reached grade 5 and grade 6, they're already under such high near-work pressure and such severe deprivation of time outdoors, it's not much different to lockdown. There's also evidence that the progression of myopia in established myopes has increased. So the conclusions: within the Asia-Pacific region, we need more data on refractive errors in order to understand just how widespread the myopia epidemic is. And what's true for the Asian-Pacific region is even more true for other parts of the world. I'd suggest it's time for a transnational project using agreed methodology for sampling and measurement in order to be clear about the current state. A minimalist approach would be to estimate unaided low visual acuity or non-cycloplegic refraction in a statistical sample of final year high school students. This will give us the best insight into what's going to happen in the future. Interpreting that data in combination with high school completion rates should give us a very reasonably accurate picture of the population prevalence of myopia.
Good day to all of you. Welcome to this talk on new genes for myopia. I'll share with you some information. To this audience, we all know about myopia caused by the elongation of eyeball, which can be due to a very simple factor. For example, just a variation in the function of a transcription factor. Or on the other hand, it could be due to a whole multitude of factors, repertoire of genetic and environmental factors. And it seems that the latter is true. We can easily replace Hong Kong by any other major cities, particularly cities in East Asia. Myopia is in fact very serious, and the trend is increasing. And for parents with high myopia, particularly concerned, will my children inherit high myopia? And the etiology of myopia can be nature or nurture. When genetics is biological, but then heredity might not be just biological. It could be due to parental lifestyle, which are inherited not through biological effects, but by physical activities. So myopia etiology can be quite complicated. If we summarize the genetics of myopia, we know there have to be heredity. Myopia parents and trim studies. Then, of course, again, our concern is the heredity can still be external. And then I put a question mark in the ethnic difference in prevalence. Are there real ethnic differences? And then, but there are solid data from primary linkage analysis and GWAS and whole genome sequencing. There are genetic factors. Then Ian Morgan's very brilliant review in 2018 will discuss the issues and also the factors, external factors affecting myopia other than genetics. I will still put a lot of question mark on the interactive additive effect of lifestyle, particularly near and outdoor activities that would lead to myopia with genetic factors. There are more than 200 reported genocidal myopia across all chromosomes. But I'd like to mention this particular paper in Nature Genetics by Paro that's summarized, which is a meta-analysis of more than 500,000 subjects of European ancestry, and the new genes and mechanisms that be summarized in this meta-analysis, and top regions associated with myopia all been stated. So this is the most updated specific genes associated with myopia. My attention is also on the gene-environmental interactions, and there are important landmark papers characterizing the association of education with more myopia and outdoor activity associated with less myopia. Very important work being published. And back to the Chinese University of Hong Kong Department of Ophthalmology and Visual Sciences, we are also among the groups searching for the myopia genes who works through population-based association studies, GWAS, and whole exome sequencing in myopia trials. And recently we have started whole genome sequencing in high myopia trials. And we work on the genetics and want to pay attention particularly to genes and lifestyle. For a few years, been working on this children eye care program, which is also a community-based study and community-based program with charitable eye clinics, public education workshops, eye care ambassador program to help the families, and we do outreach, we have public engagement, and of course there's a scientific part, that's the epidemiological database. And we are working on, and we are closing by now, completing 35,000 families. Each family, one child. Actually, some family we have more than one child for and then actually the actual number is slightly more than 35,000, and all their parents. So this is a family study, children and their parents, population-based sampling, and we want the work to be of community impact, society impact, and of course scientific and medical impact. We've been gathering some data. We have found that high myopia children have at least one high myopia parent. Of course, we know about that, but that's very clearly found in this study. And among the parents, high myopia occur in 14% of them. That's in Hong Kong, actually. Like many other, that's why I said many other major cities. High myopia is actually very prevalent, and parents high myopia have dosage effect on children developing myopia. And we also have very clear evidence that when both parents have high myopia, there are more than 50% chance of the children developing myopia. So this is very clear in the study. And we also pay attention to quantitative ocular trace, because children ocular trace, particular as time goes, if not attended, or if they develop into eye diseases, and that would lead to the deterioration of the quality of life due to the vision impairment from newborn all the way to the elderly as time ages. So we pay attention to quantitative ocular trace, and we have some data. One has been published that WNT7B and RS01 associated with axial length in school children. And there are also genetic associations of severities and endophenotypes of children in myopia, moderate and high myopia. Our work on GWAS in the association with trace and are still ongoing. So we have some data coming up, and 10 SNPs, in fact, those are reached suggestive significant level, and we are working on them. And then 10 lymphadenopathy, reaching significant level, 10 to the 10 to minus 6 of myopia progression in 1 year. And then we are now going on to 3-year follow-up on the myopia progression, and then doing the data analysis. So that's what's been going on in hunting or mapping, finding, exploring the genetic basis of myopia through all these various sorts of studies. And that's been going on in Hong Kong, and I must acknowledge all the contributions of my clinical colleagues, in particular led by Clement, the genetic part, now led by Guy, and the children program led by Jason. And we work together, trying to use Hong Kong and the John Santo International Eye Center, directed by Professor Mingzi Zhang, and our base in Hong Kong, the Hong Kong Eye Hospital, the Prince of Wales Hospital Eye Center, the CHK Medical Center, the Hong Kong Children's Hospital. And in Shantou, we are building a new hospital complex that should be operational within 2 years. So altogether, we advocate the concept of children's healthcare through eye care. Thank you very much indeed for your attention. Thank you.
Thank you for the invitation to speak at the APMS Congress. I'll be speaking on the prediction of myopia or risk stratification. The high myopia rates are very high in countries such as Korea, more than 90%, and in Singapore, more than 83%. However, they're not that high in other countries around the world. And when we look at this review, published in Nature Reviews, the rates of myopia are also high in other cities in China as well as in Taiwan. Why are we concerned with high myopia? High myopia may lead to pathologic myopia, such as myopic maculopathy or staphyloma, and that may result in visual impairment in patients with myopia. How do we know which myopic child will develop high myopia? There are certain risk factors for myopia. For example, if a child develops myopia at a very young age, that child had a higher risk of high myopia later in life. Or if a child has a higher myopic refraction at baseline, that child also has a higher risk of high myopia later in life. Parental myopia is also another known risk factor for high myopia later in life. I'd like to present the results of this study of rapid myopia progression in early childhood as an ocular predictor of high myopia later in life. This study is funded by the EDB Johnson & Johnson Vision Grant. It is a Singapore Industry Alignment Fund, and the co-authors of the study include 2 employees from Johnson & Johnson Vision. The SCORM study includes 1979 children aged 7 to 9 years. They're followed up every year for the first 8 years in the school. Cycloplegic eye drops were instilled, auto-refraction was performed, biometry was performed using the A-scan machine, and the children were seen yearly in the schools from 1999 to 2001 to the year 2007. In this study, we looked at the myopic children and we followed up the children till they were teenagers, aged about 12 to 19 years of age. And in the teenage years, we looked at whether it's about high myopia, moderate myopia, and looked at the severity of refraction or axial length. So we know that 20% of the teenagers had high myopia and 35% had long axial lengths of more than 25 millimeters. And the baseline refraction and the change in myopia progression was correlated with the final refraction in the teenage years. So what were the key factors? The 3-year change in myopia progression was a key factor to determine the risk of high myopia. The multivariable odds ratio was 11.4, alongside baseline SC with the odds ratio 11.8. And age of myopia onset was also another key risk factor for developing high myopia later in life. We see the same pattern for axial length. Which child will develop a long axial length as a teenager? That will be a child with a faster axial length progression, a longer axial length at baseline, and an earlier age of myopia onset. For teenage spherical equivalent, more myopic refraction will be seen if the child has a faster myopia progression, more myopic SE at baseline, or earlier age of onset of myopia. And we see the same results for axial length as well. In the AAC curves we see here, the combination of a 3-year refraction progression and a more myopic refraction that would accurately predict whether a child would develop high myopia as a teenager. And we see the same pattern for axial length. Thus, our data shows here again, if we looked at myopia progression, if a child had a faster myopia progression, then the child would have a higher risk of high myopia. If a child had more myopic refraction at baseline, that child would also have a higher risk of high myopia. Thus, myopia progression in childhood is a new ocular refraction parameter that can predict future high myopia, and the combination of myopia progression and baseline refraction and age of onset of myopia should be important guiding factors for determining whether the child will develop high myopia and the type of treatment that will give a myopic child, whether it will give a myopic child a more aggressive treatment or a milder treatment. So it's very important for counseling and treatment of myopic children. What about genes? In the same cohort, in the SCORM cohort, we also looked at genes as we perform a GWAS analysis of Chinese children using the Illumina chip, and we developed an Asian polygenic risk score to predict high myopia in the SCORM cohort as well. We looked at the genotype data from SCORM and combined with that of the 1,000 genomes and performed multi-dimensional scaling analysis. So we can see here in these plots the relative distribution of the Chinese children in SCORM in blue with the other studies that were pictured here. So we developed a PRS based on the very large European GWAS study, the largest thus far, quoted here by C et al. And we looked at only the genome-wide significant SNPs and those that were associated with refractive error. And we looked at the probability of a conditional association with the SNPs in the STARS Chinese study, and it had to have a conditional association of genome-wide significance. So we found 655 SNPs, and the PRS was calculated for every child in SCORM as a sum of risk alleles weighted by the effect sizes. So we see here in this histogram the normal distribution or the polygenic risk scores, and then we wanted to compare the polygenic risk scores with other factors such as time outdoors and parental myopia in determining several different phenotypes: teenage refraction, teenage axial length, time to moderate myopia, and time to high myopia. So we looked at spherical equivalent and axial length as a teenager, and we looked at the R squares and the incremental R squares. We see that the new PRS alone had a better predictor than just parental myopia alone or time outdoors alone, and we see the same pattern for axial length. If we use the Cox proportional hazard model and calculated the hazard ratios for time to high myopia, the polygenic risk scores and parental myopia had the highest hazard ratios compared to time outdoors. And for time to moderate myopia, the polygenic risk scores and parental myopia had high and similar odds ratios, and they were higher than time outdoors. Here we see the AUC curves. The curve on the left is for high myopia, and the curve on the right is for moderate myopia. And we see that the curves have a very high AUC if we combine age, outdoor time, parental myopia, and the PRS. Here again, to depict the relative accuracy of the PRS with time outdoors. And on the left, we see the prediction for high myopia. On the right, the prediction for moderate myopia. You see here again that the PRS had a higher odds ratios for both moderate myopia and high myopia compared to time outdoors. We did replicate our study in the gastro birth cohort for myopia, and we looked at the effect sizes of the PRS on spherical equivalent and axial length. And we found similar findings, and that our findings replicated in the gastro study. Thus, in conclusion, we found two important new predictive factors of high myopia later in life. The first is an ocular factor, early childhood myopia progression, and the next is a new Asian polygenic risk score, which will improve the predictive performance to detect children at high risk of high myopia. I'd like to conclude by thanking my team in Singapore and all my collaborators in Singapore and around the world. Thank you.
Good afternoon, colleagues. It is my great pleasure to share what is new in the Hong Kong Children Eye Study. I have no financial interest to disclose, and I would like to briefly introduce our study and also highlight some of the key findings in the Hong Kong Children Eye Study. And the Hong Kong Children Eye Study has three phases. The first phase has been completed in 2015 to 2018, about 5,000 families. And the second phase is a 3-year longitudinal follow-up. And we are now completing the third phase, which has increased our sample size to 30,000 children. And what I'm presenting today is mainly focused on the first phase. And so we use a cluster random sampling. The children are aged 6 to 8 years, and also their parents. We have done a comprehensive eye examination in our CHK Eye Clinic center at our hospital. So in fact, we bring the children in to our clinic for a detailed comprehensive eye examination. So we look into both the genetic and also environmental influences in myopia progression. So number one, we have determined a very high prevalence of myopia in Hong Kong, which is one of the highest in the world. Hong Kong, in the age of 6, 7, and 8 years of age, about 36% of them are already myopic. And also not just the children, we also look into the adult, their parents. We found that more than 72% of their parents are myopic, and among them, about 14% of them are highly myopic. This is, again, one of the highest in the world. And also, we look into the parental and also the genetic and also the environmental factor. In particular, the parental data, we look into the 2055 family trios. So we are the first group to have both the parents and also the children exact eye examination data to have the analysis, and we found that the parental myopia in fact confer a very strong independent effect on children myopia in a dose-related manner. You can see that the stronger, and both parents are highly myopic. In fact, the OR of having children myopia is 12 times higher. Age and the parental myopia are two important indicators to predict the rate of myopia progression. Number 3, we also look into the genetic factor for myopia and myopia progression through several publications. We look into the PASIX gene and also the SNTB1 gene was associated with extreme myopia but not low-grade myopia. We also look into the EZC 3H11B, B1CC, BICC1 gene for the moderate and high myopia, and the EZC 3H11B, KCNQ5, SNTB1, and GJD2 are risk to excessive axial length in children. And we also look into 3-year follow-up to look into the myopia progression genetic, and we found that the FHX1B, KCNQ5, and GJD2 gene are risk factor for this myopia progression in children. And we also look into environmental factor. Number 1, we look into the reading posture, the reading distance. We do the video camera to capture the whole reading process of the children, and we found that the shorter habitual reading distance are associated with a higher risk of myopia, and also among them, for those with under-corrected myopic children, they have a closer reading distance than those with non-myopic and fully corrected myopic children. We are also looking into how COVID-19 affect the children's lifestyle and affect the myopia progression. So we look into 2 cohorts, the COVID cohort, which is 700 children, and pre-COVID cohort, which is a 3-year cohort in 1,000 children. And we found that there is a significant increase in the incidence of myopia, almost 2.5-fold increase during the COVID pandemic time. And the myopia progression almost double, and also the axial length increase is almost 1.5 times increase. And this is also associated with a decrease in the outdoor time. Outdoor time has dropped to almost 0.4 hours per day during the COVID pandemic, and also the screen time has increased to almost 7 hours per day for the children during the COVID pandemic. We also look into how the lifestyle affect the children's ocular health, including one of the important topics is the secondhand smoking. We are the first group to identify that children exposed to secondhand smoking is associated with a choroidal thinning. This choroidal thinning is along a dose-related response. The larger number of family smokers and also the larger quantity of secondhand smoking is associated with a more thinning of the choroid. And not just the thinning of the choroid, our follow-up study also suggested that even the retinal nerve fiber layer are also thinning. And this is again along a concentration, dose-related response. And we also look into the children retinal vasculature. One of that is to look into the fundus picture, and we quantify the retinal vascular parameter using the central retinal arteriolar equivalentia AE and central retinal venular equivalentia VE. And we look into how the blood pressure, obesity effect on this. We found there is an independent and synergistic effect of high blood pressure and obesity on all these retinal vasculature in children. We are also the first group to look into one of the first group to report the OCTA data in children. In 1,000 children, we reported the OCTA finding, and to provide new information on the baseline morphology of all these OCTA data of the retinal microvasculature and the associated factor in children. And the follow-up study, we look into the amblyoptic eyes, in 1,000 children normal data and also 30 subjects with amblyopia, we found that in amblyopia, the children has a lower FAC circularity, lower vessel dimension, and larger vessel diameter index. These data suggest there's a presence of microvascular abnormality in amblyopic eyes. We are very glad that to have contributed to the understanding of the international literature on children myopia and also children ocular health development. We would like to thank our funding agency for all their support and also our department and all the whole team. And I would also like to thank my mentor, Professor Kelvin Pang and Professor Clement Tam for all their support, advice, and guidance on all this work. And my partner, Professor Chen Lei Gai, Dr. Gam Ga Wai, and Ms. Mandy for we all working together on all this study. Thank you very much for all your attention. Thank you.
It is my great honor to be invited to share this talk about evidence-based public health intervention for myopia. I'm Pei Changwu from Taiwan. In East Asia, there is the highest prevalence of myopia. Up to 80% of junior school students with myopia, and 20% has high myopia. As we know, the high myopia has high potential to become pathological myopia in later life. It is a potential serious public health issue. It has a huge economic burden, estimated around $250 billion every year. In WHO and IAPD combined meeting, several countries reported that there is barrier to implemented intervention for myopia, especially for lack of awareness of myopia, including in Australia, Cambodia, Malaysia, Mongolia, and Vietnam. Unaided visual acuity is widely used, a simple, useful method to detect the myopia because it has high correlation with myopia prevalence. In Taiwan, school nurses perform visual screen every semester for school children. Recently, Professor Morgan and the researchers reported that two major factors, including education and time outdoors, are strong and causal relationship with myopia. Several large cohort studies, including Syrian myopia study, Orinda study, Singapore cohort study, showed that outdoor activity reduced the prevalence of myopia in children. The higher levels of evidence, including two large RCT randomized trials. One is performed in China, Guangzhou, by Professor He Mingguang and other researchers. And the other large RCT study is performed by our team. Both studies showed that additional time outdoors has positive effect for preventing the myopia onset, reduce the myopia incidence. In addition, time outdoors also have effect on myopia control, around 30% efficacy. The effect also could achieve even with moderate light intensity, such as in hallway or under a tree. Outdoor time could prevent myopia progression. Several meta-analysis studies confirm that outdoor activity is a protective factor for myopia onset. A study showed that every additional hour of outdoor time per week could decrease the odds of myopia by 2%. In 2010, Taiwan's Ministry of Education accept the suggestion of increased time outdoor to implemented in Taiwan school children vision care program. It named the 120, means that an additional 120 minutes per day outdoor time was intended to prevent myopia. This figure shows that the prevalence of reduced visual activity in 1 million school children in Taiwan. We can see before 2010, there's a trend of increased prevalence, but after the 120 outdoor time promotion, the trend reversed and continues to decrease every year, around 1%. In contrast to outdoor activity, the other important risk factor is near work. Even though there was lack of large randomized clinical trial for near work intervention, but we have done a meta-analysis to study the association between the near work activity and the myopia. The result shows that every 1 diopter hour near work per week would increase the odds of myopia by 2%. Another 2 important risk factors, including education and screen time, both has high correlation with near work, but due to still lack of the large study and the randomized trial, maybe further study and research needed to confirm and propose effective intervention method to reduce the impact of these 2 important risk factors. Not only in Taiwan, the public health intervention with outdoor time for myopia also occur in other countries, including in Singapore. The Ministry of Education also allow that require preschool children to receive up to 1 hour outdoor time daily. The researcher also recommendation to the government that the 2 to 3 hours per day outdoor time may be needed. In China, the Chinese state government set targets to reduce myopia in recent years. They focus on changing the behavior such as increased outdoor time and parent education. In conclusion, public health intervention should be supported by evidence-based. For the myopia, prevention is important. At first, we needed to recognize myopia and high myopia as an emerging public health issue, and advocacy to the government to give even higher priority to fight myopia. National vision survey and data collection are important because we needed to monitor the myopia prevalence and its trend. Even though the increased time outdoors for myopia intervention has strong evidence, but implemented to public health policy is more important, such as refined curriculum in school health program to include extended outdoor time. Myopia intervention and control is not easy. It needs multidisciplinary cooperation, including parents and teachers groups, public health workers, medical professionals, eye practitioners, and NGOs, and also cooperated with WHO office to have more advocacy, education, and set up guidelines. Thank you for your attention.
Hello, everyone. This is Ming from Melbourne Unit. So first of all, thank you very much for the opportunity to present. Today, I'm going to share with you one of our data simulation study where we try to estimate what would be the intervention strategy might change the prevalence of myopia or even severity of myopia in the population, in particular in the context of ex-Asia population. So as most of us sitting here would know, the increased time outdoor has been proved to be an effective protective factors for myopia onset in particular in both clinical trial in Guangzhou, Taiwan, and a number of other studies. So then in Taiwan, Dr. Pei Tang Wu's group actually further proved that increased time outdoor actually also is really because of the light intensity or sunlight exposure. We not only reduce the myopia onset, but also change the endpoint, or say like slow down the myopia progression in the intervention arm. So Pei Tang's group in Taiwan also demonstrate interesting population prevalence of myopia trend in Taiwan. So they use uncorrected visual acuity less than 20/25 as a surrogate for myopia. And then they observed that myopia prevalence actually increased over time from 2001 to 2010, despite all these active myopia intervention strategy to around 50%. So, but then once they initiate the outdoor activity or outdoor intervention, for in particular, the 2 hours outdoor intervention, and also every 30 minutes, 10 minutes break for the near work, they observe a prevalence of myopia reduced from 50% to 46% in the Taiwan population. So in China, based on the 12 years follow-up data in the twin study, we kind of demonstrate that a delay on the age onset of myopia will actually quite have a dramatic impact on the we call it endpoint refraction when people reach adulthood. For example, if demonstrate age onset, if 8 years old, age onset of myopia is 8 years old, and then it will end up -6 or greater diopters. But then if this is if the age onset is 15 years old, it's much lower. So we also construct the model to estimate what is the endpoint, refraction endpoint, when in the context of different age at onset. So in this study, we particularly interested to look at what is the long-term effect of increased outdoor time as a myopia strategy. We try to understand how this might change the prevalence of myopia in the population, and also try to understand how this might change the degree of diopters at adulthood, like when the children or people turning 15 years and older, using three study data. So first of all, we set the reference population. We actually used the IESC study conducted in Guangzhou in 2004 as to set a reference population, which means without intervention, what is the prevalence in the population? So this, as you can see, from 2.7% in 6 years old to increase to 73% in 15 years old. So then we use the myopia instance data from the Guangzhou Twin Eye study. Like this is the instance data in different age group. This is very similar actually to many other longitudinal study conducted in the ex-Asia or Chinese population. So then we use the data from the randomized from the outdoor clinical trial to estimate what's the prevention strategy, prevention effect. For example, for 7 years old, without the intervention, the instance is 8.7%, but then with the intervention, this will be reduced to 5% instance rate. So we're representing a relative reduction of 42.5% reduction on the instance rate. And then similarly, among 8 to 9 years old, in the clinical trial, we demonstrate around 30 or 14% of the reduction on the instance of myopia. So now we look at the reference, what happened in particular in the prevalence of the myopia in the population. So this is a IESC prevalence data without intervention. So this is instance relative instance reduction in different age group. So we calculate it, and then we assume that because there's no intervention in the preschool population, so the prevalence of myopia among age 6 would be unchanged, it remain the same, but then the intervention is in the primary school, like from grade 1 to grade 6. So we'll reduce the prevalence of myopia in this population. And then eventually, when we first year intervention, if we intervention second year, third year, fourth year, and up to 10 years. So after 10 years intervention, the overall prevalence of myopia will be reduced from 34 or 35% to around 30%. We're talking about 5% reduction on the prevalence data or the prevalence in the population. So then we also try to estimate what is the impact on the refraction diopters at 18 years old, 15 years old, sorry, 15 years old. So we estimate that the in the IESC study cohort in 15 years old, among the high myopia patient, actually the mean diopters is minus 7.1 diopters. And this is the age onset data that I already shared with you in the Guangzhou twin eye study. So we demonstrate and then we construct a model, and then you can see if there's different age onset, the endpoint refraction data, refraction at 15 years old, very different. Now we are talking about different intervention strategy. If we have a 6-year intervention, which means the intervention will be implemented in all from year 1 to year 6, grade 1 to grade 6 in the primary school, and 3-year interventions, the intervention only in the early, like a younger group, like from grade 1 to grade 3, and 4 years intervention, we extend the intervention from not just grade 1, but also preschooler, like 5 years old also. And 3-year double effect, we assume the preventive effect will be double, the intervention effect will be double. And what happened on the prevalence of myopia? Actually, we demonstrated this is 5-year intervention, this is a 10 years intervention. It will reduce the prevalence of myopia in the population from, if it's a 6 years intervention and 3-year intervention, it doesn't change the prevalence that much. But then if it's a 10 years intervention, I think actually the 3-year double effect intervention got a much better impact in terms of reducing the prevalence of myopia in the population. So as you can imagine. So then we also, this is a figure showing, how this intervention, 6 years, 3 years, and also 3-year double effect will change on the prevalence data in the from age 6 to age 15. As you can see, of course, the message is 3-year and 6 years intervention doesn't change the prevalence of myopia that much, very similar effect actually, but then 3-year double effect, of course, have a much better reduction on the prevalence of myopia data. So more interestingly, if we look at the degree of refraction or refractive status among the high myopia patient in 15 years old. So with the 5 years or 10 years intervention, the 10 years intervention, after 10 years intervention, actually we are able to reduce the refraction from minus 7.1 to minus 3. So given the fact that as demonstrated by polymer study, they demonstrate every single diopters reduced on the myopia will reduce the likelihood of developing myopia maculopathy by 40%. So even a 2 diopters reduction on the refraction will have a tremendous effect on reducing the chance of developing myopia maculopathy. So in conclusion, outdoor activity are quite effective in reducing prevalence of myopia in the overall population. So 6-year intervention and 3-year intervention, similar effect. But then if you extend the intervention to preschooler, that you will see a much better myopia prevalence reduction. So the intervention with double effect, if this hold true, will significantly reduce the population prevalence as well as the degree of myopia in adulthood. Thank you so much.